So far we have looked at the citric acid cycle in a very narrow scope as a means to provide reducing equivalents for the mitochondrial electron transport chain. And the goal of today, the last part of this lecture, is to look at the citric acid cycle in more wider context of cellular metabolism. Almost every single intermediate in the citric acid cycle can be also precursor for some anabolic pathway, as was discussed in the first part of this lecture. Citrate is used to produce lipids. Alpha-ketoglutarate can be used to produce a number of amino acids and purine type nucleotide bases. Succinyl-CoA can be used to produce porphyrines. Oxaloacetate can also be used to produce a number of amino acids and pyrimidine type bases, and this is the only small fraction what can be produced from the citric acid cycle intermediates. And this raises a very important problem: how the intermediates could be replenished to run smoothly the reaction of citric acid cycle. And the problem is that, as we discussed, when the acetate is oxidized through the citric acid cycle, there is no gain or loss in the intermediates. Everything which was pumped in the form of a steel group in the citric acid cycle go out in the form of carbon dioxide and reduced electron transport carriers. So for this anabolic pathways stemming from citric acid cycle to take place, so there must be a way to replenish the intermediates of the citric acid cycle, and this is accomplished through a network of biochemical processes called anaplerotic reactions that replenish essential intermediates of the citric acid cycle so that it could act as an amphibolic pathway, working both ways, as in catabolic fashion and in the anabolic fashion. So there are a number of anaplerotic reactions, but majority of them focus on replenishing the last metabolites of the citric acid cycle, oxaloacetate and malate. The 4-carbon compounds using 3-carbon compounds as the precursors. And in fact, we are familiar with some of these anaplerotic pathways through our prior discussion on the glycolysis and gluconeogenesis. So there are several pathways to replenish oxaloacetate. One of them uses pyruvate, bicarbonate, and ATP as a precursor in the reaction, which is essentially the same as the first bypass step in the gluconeogenesis pathway using pyruvate carboxylase. So in this generates oxaloacetate that can be fed into the citric acid cycle. Another way that produces oxaloacetate is the reverse of the second step in the first bypass of gluconeogenesis. There, the phosphoenol pyruvate is carboxylated, and this is coupled by substrate level phosphorylation, giving rise to energy in the form of GTP, and also generates oxaloacetate. There is a shortcut of this pathway where no substrate phosphorylation is produced, and oxaloacetate generated by carboxylation of phosphoenol pyruvate using the bicarbonate and releasing inorganic phosphate. So the exact flavor of the anaplerotic pathway, the preference is dependent on the tissues and then on the organisms. Some organisms have also pathways to regenerate malate by concomitant carboxylation of pyruvate and its reduction using the bicarbonate respectively and the reduced form of NAD or NADP as the reducing agents, thus producing malate and regenerating oxidized form of the respective electron transport carriers. The malate regeneration pathway relies on malic enzyme, which is not present in our organism, but is this pathway, anaplerotic pathway, is widespread in the biosphere. While metabolite availability, intermediate availability, is the measure way of regulating the metabolic flux partitioning between anabolic and catabolic mode of citric acid cycle. The regulation of the metabolic flux through the citric acid cycle to adequately satisfy cellular metabolic needs is accomplished by different mechanisms. Similar to glycolysis and glyconeogenesis, the steps that regulate that are used to regulate the flux through the citric acid cycles are the ones that have a highly exergonic at cellular conditions, and this is easy to understand. If cells undergo the metabolic interconversions through highly exergonic steps, it cannot be reverted back. It is a unidirectional process. This way, this is commitment step, and this is an important point where the flux through the pathway could be regulated. As such, within the framework of a citric acid cycle, there are 4 steps that are highly exergonic and hence regulated. This is the conversion of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex, the condensation reaction of citrate synthesis by citrate synthetase, and 2 oxidative decarboxylation steps within the citric acid cycle where the carbon dioxide is evacuated, chopping off the carbon skeleton of the citric cycle intermediates. The regulation of the metabolic flux is achieved in 2 fundamentally different ways. First is a direct way where the metabolic flux is regulated by the substrate availability and the product buildup. If there is a lot of very little substrate or there is a lot of product, then the mass action term of the reaction energetics will compensate for the highly exergonic nature of the reactions, thus stopping the net flux through the pathway. Another is indirect way by regulating the respective enzymes that commits these steps through mechanism of allosteric regulation. And to understand how the enzymes are regulated, it is practical to address it from the perspective of cellular needs. Cell needs are the high flux through the pathway if the there is a build-up of the precursors necessary for running the respective steps, and when there is shortage of the final products. Likewise, it makes sense to inhibit enzymes and inhibit flux if there is a final product build-up uh, or there is shortage of the respective precursors. So, and considering that, one can deduce the uh, uh, inhibitors and activators of the respective regulatory enzymes sitting on the um, four regulated steps of the citric acid cycle. Cells would like to activate the flux through citric acid cycle if there is shortage of ATP. Not surprisingly, several out of these steps are regulated positively by AMP or ADP, the products of ATP hydrolysis. Likewise, the metabolic flux has to be inhibited if there is a lot of ATP in the system. And unsurprisingly, the ATP is an allosteric inhibitor for several of these enzymes. Curiously, calcium ions are activators for several of these enzymes, which is practical in the case of skeletal muscle, where a high concentration of calcium in cell cytoplasm signals high activity of sarcoplasmic reticulum, which activates actomyosin, which in turn require a lot of ATP for contraction. So this would generally signals the need of more ATP and high metabolic flux through the pathway. So, but if one would have build up of the intermediate products such as the reduced form of electron transport carriers, this would signal that the flux through the pathway has to be down regulated, and then one could see that the NADH, the reduced form of the electron transport carrier produced by citric acid cycle, is a negative regulator in some of these steps. So there are more complex way in the regulation of these regulatory enzymes. For example, the pyruvate dehydrogenase complex, its E1 subunit is phosphorylated by a specific regulatory kinase, which is activated by the high availability of ATP, thus blocking the flux through this pathway, and there is a respective phosphatase which reversibly activates the enzyme. So there are some metabolites that are master regulators that coordinate the flux through several different pathways. For example, citrate is not only inhibitor of citrate synthetase, but is also inhibitor of phosphofructokinase and glycolytic pathway that signals that there is a build-up of citrate at some stage of citric acid cycle. So there is no need for pyruvate to be produced through the glycolytic pathway. And this is only a small fraction of how the metabolic flux is regulated. So at the end, one could think of the regulatory enzymes that sit on the top of these regulatory steps as the signal integrators that sense a lot of positive and negative signals generated by other parts of the metabolic pathways to adequately regulate the flux through the citric acid cycle. So altogether, we have looked at two major steps of cellular respiration, production of acetyl-CoA and citric acid cycle. We looked in detail at the production of acetyl-CoA could appreciate that it is catalyzed by a very sophisticated multi-enzyme pyruvate dehydrogenase complex that requires a number of cofactors and works as an enzymatic factory. We looked at the catabolic nature of citric acid cycle, looked at the chemical logics of its steps and how it is used to produce not only ATP and GTP direct energy equivalents, but stockpile of electron transport carriers in the reduced form to feed electron transport chain. We looked how the anabolic and catabolic nature of citric acid cycle could be partitioned through the supplementation of metabolites, intermediates through the anaplerotic reactions, and how the citric acid cycle is regulated through the metabolite need and availability to adjust to the cellular needs.